board. And we'll go ahead and put this up on display. All right, so on Monday we were talking about our genetic code, and let me see if I can just back up. So we had talked about how many of the nucleic acids in the DNA itself, or the nucleotides in the DNA itself, were part of this protein coding region. All right, we talked about that being a really, really small percentage. And so that if mutations occur, right, it's, you know, a really small piece of this puzzle. And so oftentimes if there is a mutation, it's not really going to impact the functioning of the cell. All right, but what we're going to do now is we're going to look at what happens when we have these mutations in those protein coding regions. All right, so in that 1.5%, and we're going to look at these different types and the impact that they'll have on the functioning of that protein. So this slide is showing us 3 different versions of the same gene. Okay, and so the one here on the left is the normal non-mutant, sometimes referred to as the wild type gene. And so we have our DNA double strand here, and this would be our template strand, right? And so the transcripts, so the red here would represent then that messenger RNA strand, and then here are the amino acids that are linked to these specific codons. And so we can see that we have proline, glutamine, and glutamine here as our 3 amino acids, and this particular gene would function normally. This is the non-mutant strand. One type of mutation that can happen is known as a silent mutation. And so if we look at this particular nucleotide here in our DNA strand, we'll notice that this is different than what was in the wild type or the non-mutant strand. So we had a GC nucleotide pair here, where now we have an A and a T. So these are paired correctly, but this shouldn't normally be an A, right? This should normally be a G. And so what we see happen here is we still have the same 3 words, right? So we still have, you know, we haven't changed our sentence, and we'll look at those types of mutations in a little bit. So we still get this proline. We didn't change this one at all, so it still says glutamine. But because of the redundancy that we saw in that table the other day about how different codons can code for the same amino acid, we see that this one mutation here doesn't actually change our end product. So our protein here is going to be the same. So that's why we call this a silent mutation, because we don't see this. So you would have to have your DNA tested to find this one single point mutation to even know that that existed, because all of this protein would be functioning just as normal. No one would ever know that this happened. Now, the next type is known as a missense mutation. And so again, we are looking at a point mutation, so just one nucleotide change in our DNA strand here. This time, it looks like we've flip-flopped the A and the T here, right? So this used to be AT, and now it says TA. All right, and so let's see what happens with this type of change. All right, so now we see down here in our messenger RNA strand that we've changed the A that was normally here to a U. So now we have GUG. So if you go back and look at the codon table, we'll see that GUG no longer codes for glutamine, it now codes for valine. All right, and so now we do have a change in our protein structure. This would be an actual mutation that would show up, and this may cause the protein to misfunction. Okay, so we do end up with an actual change here in this protein. Okay. So other changes that we can see happen. This again is our non-mutant form. And then, oops, I forgot to put the title here. This mutation is actually now going to be what we call a nonsense mutation. So what happens here is that we see another change. So in this situation, the GC has been changed to a TA, and it's still a point mutation. Point mutation meaning that I've only changed one nucleotide. And what happens now is that mutation actually caused a UAG codon, which is a stop codon. So we wouldn't even be able to finish transcribing the rest of this protein because this mutation inserted a premature stop codon. So nothing downstream from here is going to be able to be transcribed. So these nonsense mutations cause a lot of havoc in protein function, right? Because we can't actually build our protein. Right, so those are kind of our three types of mutations. We have the silent mutation, which doesn't alter the protein, the amino acid sequence, so our protein remains unchanged. We have a missense mutation, which does cause a change in the amino acid sequence of our protein, which can lead to a misfunction of our protein. And the last one is a nonsense mutation, which inserts a premature stop codon, and this definitely will impact our protein formation and give us a non-functional protein. All right, so let's look at what happens when we kind of put these mutations in different places in our protein sequence. And so we're going to look at this by using three-letter words that make a sentence in the English language. All right, so if we pretend that the big boy saw the cat eat the bug is our genetic code for our protein, and this is the correct sequence of those letters, we can see here, well, I'll have you guys, what happened in this first example? Where did the mutation occur in that first sentence? Does anybody see where the mutation is and what happened? Right, the E is missing, right, from the word the. So everything shifted kind of one spot to the left. And so you can see how when we have a mutation that occurs at the beginning of our protein sequence, or our, what do I want to say? Maybe at the beginning of our messenger RNA transcript there, if it occurs here at the beginning, it messes everything up, right? This doesn't even make sense. How about in the second one? Where does the mutation occur here? And what is the mutation? Did we add a letter, delete a letter? And where did that take place? Does anybody see it here in the second example? All right, so there's an extra T, right? Right here. So we added an extra T in the second the. So the first part of our sentence still makes sense. We have the big boy saw, right? But now I've altered the remaining part of this transcript, right? So each of these three-letter codes would give us a different amino acid than the original strand up here, right? And we don't even finish our sentence. We've lost one of them. All right, so the point of this is maybe to write down or emphasize in your notes that the earlier in the transcript that we have the mutation, the larger the impact that mutation is going to have on the rest of that sequence. All right, the later in the transcript where the mutation occurs, the less impact it may have. All right, and this type of mutation is an insertion or a deletion. All right, so it's still just dealing with one of the nucleotides, but this time we're either deleting it or we're adding it, and these can cause a lot of chaos in forming our proteins. All right, so if we look at, here's a couple more examples of insertions and deletions. So an insertion adds a new nucleotide to our sequence, where a deletion results in the removal of a nucleotide from our sequence. And when we have these insertion or deletions, what we call this is a frame shift. Okay, so down here in the left-hand corner, it's a frame shift, meaning that, you know, I've shifted all of the words now in that sequence, right, as I'm reading that in those 3-letter codes. So if we look at the one here in the upper left-hand corner compared to the one down here in the right-hand corner, which one of these represents a insertion and maybe which one represents a deletion? Does anyone see what those differences are? I hope I put them in here correctly. So what do you think this one here on the left is? Can anyone tell what happened with this red one? So it kind of looks like we have an insertion there, right? So this wasn't originally here. So this is an additional nucleotide that got added. And so when we do that, we have a change down here. All right, so how does this look with our frame shift mutations? So at the top, we have our wild type, our non-mutant DNA strand. In the next one, we have your transcript again, so your messenger RNA strand. And then we have in the bottom here our protein, right? So our amino acid sequence. Now, because we have that insertion that we just saw on the previous slide, we've inserted a new nucleotide into the sequence. And you can tell it's at the very beginning. So it was in the 4th nucleotide position. So that first nucleotide came out the same. So the proline in this mutant is the same as the proline here in the non-mutant. But when we look at the second amino acid, now we see that they have all shifted, right? We don't have anything down here that matches what is up here. So this is going to give us a completely different protein than what was intended. Okay, so hopefully this shows you the impact of just a single nucleotide being inserted into the DNA, right, as a mutation, that it's going to cause a huge change in what that protein is going to look like and be able to do. So here's a question. If we deleted 3 nucleotides or inserted 3 nucleotides, would that result in a frame shift mutation? Meaning, would we see such a, you know, a drastic change in the amino acids that get created through this process? What do you think? Okay, so we have someone saying yes, that we would technically see it. You would just have an extra amino acid. Yeah, so we are going to have an extra amino acid, and the rest of the amino acids, right, should be fine, right? So instead of just adding this CG, if I added 2 more nucleotides right here, it would basically insert, you know, an amino acid here, and then the rest of them should look the same as here. So actually adding mutations in groups of 3 would actually cause less of a mess than just changing 1. Great job, you guys. All right, so here's just kind of a summary of what those mutations look like. So again, in the top here, we have our original wild type DNA. All right, so we have the messenger RNA transcript, and we have the original polypeptide amino acid strand, right? So this is what it should look like. In the next one, again, was that silent mutation. So even though we changed this T to a C, we see that we still end up with the same amino acid, so nothing down here changed. A missense mutation, so here the mutation did cause a change in the type of amino acid that was inserted, but it didn't change the rest of the amino acids downstream. So this may be beneficial to the organism. It might do nothing, or it could still be pretty bad, right? So it could cause that cell not to survive. A nonsense mutation is the one that inserts that premature stop codon, and this is usually, like I said, pretty bad. So it will oftentimes cause life to not function depending on what the role of that protein was, or cause some kind of genetic disease in that individual. And then we have the frame shift, and again, this is always usually really, really bad because you're not even creating, you're creating a different protein, and so who knows what that might do. Okay, so that's a nice little summary table for you guys. So you might want to take a look at that one as you go along. And I did see there is a mistake. I don't know if anybody else caught it. So they put a C in here, and this is in your textbook. What letter should this have been? Yeah, it should have been a G. All right, so just make a note of that. That is a typo, it looks like, here in our textbook. All right, so here are just another example when we think of mutations in evolution. There are things that we can consider, right? So a mutation could be really bad, harmful. You know, it may cause that particular organism to not live. The mutation may be neutral in that it has little to no effect on the organism, or it may be beneficial. So maybe that mutation increases what we call the fitness of the individual. Fitness, not that, hey, I've got bigger muscles and, you know, I'm healthier than you, but more that it gives an advantage to that organism over other of the same species, right? So maybe that mutation gave them a better adaptation. So these beneficial mutations is actually what has given rise to all of the various diversity that we see, right? So not all mutations are bad, I guess is the point. And so you can see here, these were just mutations to the word gone. And so showing, you know, that you can mutate it so that it forms something maybe that makes sense. So we just kind of highlighted some words that use the same 4 letters, but maybe deleted or replaced a letter, and you can still get a meaningful term. All right, and then here's a real example. You don't have to memorize these specific genes, but this is looking at the gene alpha-1AT that is part of, that is codes for an enzyme that works with another enzyme called elastase. And so elastase is used in our lungs, right, that helps them to expand and contract. And so elastase actually breaks down the elastin in our lungs. So elastin is a protein found in our lungs. And so what happens in a normal individual, this gene alpha-1AT gets expressed, and we'll see that it will inhibit this elastase. And so a normal lung elasticity is maintained by this balance between elastin production and also its destruction. So you don't want to have too much of that protein. And so this gene is helping to keep that balance in place. Now, smoking can have an effect on this gene, all right, and it can cause a mutation, right? And so now we lose that balance with the elastin protein, because now we have these active elastase enzymes. And so what happens is it can cause patients to suffer from what's known as emphysema, and that's due to the result of too much breakdown of that elastin protein in the lung, which results from smoking the cigarettes. All right, so this is definitely a disease that affects many people, makes it very difficult to breathe. My grandpa actually had this, and it basically destroyed his life. So if you know anyone with emphysema, it's very, very difficult to breathe. Another one is this mutant form of the alpha-1AT gene called PIZ, all right? And so at low levels of activity, we also see this similar activity with a cigarette smoke. So you have more of this active elastase enzyme, which doesn't allow for the balance of that elastin protein, which can result as well in emphysema. All right, and another mutation actually has to do with malaria and hemoglobin. All right, so malaria, you guys are probably aware of, you've heard of this before, I'm sure, is a mosquito-borne illness that can cause people to become very sick, right? So fevers, fatigue, vomiting, headaches, and I think it can actually lead to death if not treated, especially if patients are not in good health to begin with. So what we see is that the parasite, this malaria parasite, lives out part of its life cycle in the bloodstream of humans. And so some of the alleles of hemoglobin are actually protective against this malaria. So whoops, I went the wrong way. And so we see that depending on the mutation that occurs, that sometimes the mutation in the hemoglobin can actually be beneficial to people with malaria. And so where we see this is people who are heterozygous for the mutation in hemoglobin. So they're kind of carriers of the disease for sickle cell, but they don't exhibit the full-blown effect of sickle cell disease. Those patients or those individuals actually have immunity against malaria. So we see a lot of this in the African population. So some of those individuals who are heterozygous for sickle cell, it's actually a protective thing for them because it protects them against malaria, having this mutated form of hemoglobin in some of their blood cells. So again, not all mutations are necessarily bad or good, and it can depend on the circumstance. So if you live somewhere where malaria is pretty prevalent, having that mutation would be advantageous for you. All right, so that basically concludes the lecture from the other day. So I'm going to stop my screen share there. And then we have plenty of time. So I'm going to start our next lecture, which has to do with transcription. All right, so this is lecture 22 for those of you following along. So I'm going to go ahead and share this PowerPoint with you all now. Oops, let me just get our chat back up and we'll start here. All right, so transcription, as we talked about the other day, is really the step going from DNA to RNA, right? So we're going to stay in the same language, talking about nucleotides. And basically, we're going to copy the directions to make our specific protein. So this is the area that we're going to focus on today and Friday. All right, this is found in the first part of chapter 17. You guys can ignore this. I forgot to delete it. That was from last semester. We would have done it this semester, but we're not in class together. So you can just ignore that for now. All right, so what we're going to look at today is we're going to list, again, the 3 most common types of RNA and look at their functions. We'll see that they're all important as we work through the central dogma. Then we'll spend a little bit of time looking at the initiation, elongation, and termination steps of transcription. So really focusing heavily there on section 17.1 in your textbook. And then in 17.2, we're going to look at the differences between these 2 terms, introns and exons, that are found in eukaryotic messenger RNA, and kind of look at this process of how we can get multiple genes out of certain sequences, and how we get rid of these introns during the processing of the messenger RNA. So yes, the step is pretty simple. We go from DNA to RNA, but before the RNA can actually leave the nucleus, we do have to do some modifications. And one of them is removing these introns. And the other modification that we have to do is the addition of this 5' cap and this 3' poly A tail. So these are going to be found in section 17.2. All right, so where we are in our roadmap is really for this lecture, for this particular PowerPoint. Like I said, it will probably extend into Friday, is we'll look at how DNA is transcribed into RNA, and then how in eukaryotic cells we process that RNA. And then 17.3 to 17.5, this will be our topic next week when we come back. All right, so again, just a reminder, proteins, right, are the stuff of life. We talked about that proteins are the most abundant of these biomacromolecules that we see in living organisms. And basically what we're going to be talking about here in chapter 17 is how we're able to make them. All right, so proteins come from the instructions that are encoded in that specific organism's genome. And so there's lots of redundancy between organisms. So we've talked about how this genetic code is pretty universal between organisms, that we have the same 20 amino acids. Those 20 amino acids, when put together, form these different proteins. So there are portions of this that will be the same among both prokaryotic and eukaryotic organisms. And so like I said, today we're going to focus on the transcription end, which is basically copying that particular set of instructions, that gene, from the DNA into RNA, so that I just have that specific piece of DNA that I'm interested in. And then next week, we'll look at the translation end of the process, where we now take that messenger RNA and send it to the ribosomes to actually start manufacturing the protein. Right, so one of the things that tools that we need in order to do this is a specific enzyme. So in this case, we're going to use something known as RNA polymerase. And you'll notice this is plural. There are different types of RNA polymerases, just like we saw there were different types of DNA polymerases. These RNA polymerases will be used to synthesize or to make our messenger RNA, right? And instead of using DNA nucleotides, we're going to use ribonucleotides, and it looks like I have a typo there. Apologize. So we're going to use these ribonucleotides, right? So they'll have the ribose sugar, and the thymines will be placed with uracils. So they'll be slightly different, right, than the DNA counterparts. We're still going to use complementary base pairing. We will use one strand of the DNA in order to code for the particular messenger RNA strand that we're interested in. And again, we're still going to synthesize in that 5' to 3' direction, meaning I have to keep adding things onto the 3' end. All right, so this is very similar to what we saw with DNA synthesis. All right, so this is kind of a busy illustration, but let's just kind of take it one step at a time. So up here in the left-hand corner is our entire genome represented, right? It will continue in both directions. And the top strand that they're showing us here that is running 5' to 3' is known as the non-template or sometimes coding strand. I like to refer to it as the non-template strand. That's not the one we will be using to make our RNA. Rather, we will be using the one down here on the bottom that's running 3' to 5', right? And then that way you can see I can build my RNA strand from the 5' end to the 3' end. So we'll keep adding our nucleotide bases over at this end of the RNA molecule. Since I'm working with RNA, and RNA polymerase does not require that there be a 3' end available like the DNA did. I don't need a primer here. So we are just going to be building an RNA molecule right from the start. All right. In this second picture here, this yellow strand, this is representing this portion up here. So down here's that DNA template strand. And you can see that it's running 3' to 5'. And what I'm doing here is adding the corresponding ribonucleotide to the complement on the DNA. So here I have my guanine. I'm going to attach a cytosine with my ribose sugar, right? And then as we come down here, if we look at adenine, normally I would have placed a thymine here if I was building DNA, but rather I used the uracil, right, because I'm using RNA. All right, and so we'll continue building this, continuing down in this 3' end. So it's showing us how we're using the hydroxyl group here. Hey guys, watch the chatter.
Like
Can you mute yourselves, please? Let's see. I don't know if I can.
I don't want to read a book or write a journal.
Guys, please stop talking or mute yourselves. Thank you. All right, so here's our messenger RNA. So it will form, you'll notice here the hydrogen bonds temporarily so that we can hold this in place as we are building our messenger RNA strand. All right, so here's just a quick overview of what we're going to be looking at in transcription. So the RNA polymerase is going to perform that template-directed synthesis like we just saw in that 5' to 3' direction. As I mentioned, this does not require a primer to begin, so that's different than what we saw with the DNA polymerase. In bacteria, they only have one RNA polymerase, but eukaryotes we're going to see have 3 distinct types, polymerase 1, 2, and 3. All right, so just some basic differences there. What we're going to focus on now is the initiation step. And notice from the title of this slide, it is focused in bacteria. Okay, so when people started studying transcription, they typically use the prokaryotic cell to start looking at things. And so we're going to use bacteria as the example here. And I will point out differences as needed as we go through these processes. All right, that's different between these prokaryotic cells versus our eukaryotic cells. In all cell types, initiation is the first phase of transcription. So what does initiation all entail? All right, so one thing to point out is that the RNA polymerase, so that enzyme, it can't initiate transcription all on its own, right? It needs something to tell it that the cell needs to make a particular protein. And so it doesn't just attach randomly to pieces of DNA and start, making a messenger RNA transcript. It has to be directed somehow. And so in bacteria, this is directed by something known as a sigma protein that has to first bind to the DNA molecule to let us know where to begin. And so once that sigma protein binds, we'll see that the RNA polymerase will join with it to create something known as a holoenzyme. All right, and so this is a large complex, and we'll see a picture here in a second. The RNA polymerase then we'll see is the core enzyme because this is where the active site is located that will allow us to add those ribonucleotides in the correct order. All right, so here's what this kind of looks like. All right, so we have a strand of DNA here. All right, so how do I know where to start binding? So what we're looking at in this picture is actually this gray molecule right now. All right, so this gray molecule is that sigma molecule that I just referred to that will bind our RNA polymerase. So this is kind of like a cross-sectional view of this enzyme. And we can kind of see down here is where the active site is located. So these two things need to come together before they can bind to this DNA molecule, all right? And we'll talk about some of these regions up here. So obviously this is important that I've got this highlighted up here. We'll get to that in just a second. All right. So how does sigma then know where to bind on this DNA segment? This is known as the promoter region. All right, and we will see that promoters are required for both bacteria and eukaryotic cells. And you can think of a promoter as the region of DNA that basically says this is where you need to start to basically copy down the instructions to make a specific protein. And so there are different types of sigma proteins that bind to these promoters that have slightly different DNA-based sequences so that it kind of knows, where to bind to generate a specific gene or protein. All right, so basically along, if you think about how long the DNA strand is and that there's different regions of that DNA, right, that code for specific proteins, you can kind of think that there are different sigma proteins that recognize certain regions of that DNA that will initiate then the process of transcription. All right, so this process then allows organisms to activate only certain genes in response to some type of environmental change, either internally within the cell or externally outside of the cell. All right. If you go on to take cell biology, you guys will spend a lot more time looking at these sigma proteins. It's not exactly a one-for-one. There's several different types of sigma proteins, but it's not like there's a specific one for each gene in an organism's genome. There'll be classes of them, but we are not going to go into that much detail here in 1801. But just know that these sigma proteins can recognize these certain activation sequences in the DNA molecule. All right, so if we look at the promoters in the bacterial cells, we'll see that these regions are about 40 to 50 base pairs long. So 40 to 50, A, T, C's, and G's. But these are very specific. We'll see that at 10 base pairs before the gene, that's what this -10 means. So if we pretend that bacterial promoters here, let's just pretend that this is our DNA sequence, we would find 10 letters back this way to your left here, we would find something known as a TATA box. All right, and this TATA box, again, is upstream of what I'm trying to transcribe. So this is a key to the sigma molecule, that this is where it needs to bind. And so usually this sequence itself is about 10 base pairs long. When we talk about downstream, we're going to be looking like, it would be like to the right. Again, if we pretend bacterial promoters, there's my DNA strand. Downstream things will be, to the right of that. All right. And we'll see that we also have something known as a TTGACA box that is about 35 nucleotides to the negative, so again to the left direction, that also helps to direct where this promoter needs to start. All right. So coming back to this picture that we looked at a little earlier, but now looking at more specifics about this promoter region, we're going to see here, let's start with the right-hand side. So where we see the C and the G here, all right, this is representing the very first nucleotide of the transcript that will start to generate into messenger RNA. So that stuff will be all heading towards this right direction. All right, so our transcript is over here. All the stuff that we're showing here, this is known as the promoter. So if you count backwards, this is that TATA box that is located, -10 upstream from where we're going to start. And if we keep going, -35 nucleotides from our start position, we will find the TTGACA region. Right, so this is all upstream of our actual gene. All right, so where this G would be located right here, and then this blue and purple is represented here. So this TTGACA at the -35 position and this TATA box at the -10 position is going to be found in nearly all of our upstream from nearly every transcript that needs to be formed. And so this is how the sigma knows where it's supposed to bind. So if you look, the beginning of that sigma molecule here is binding near where that -35 box is, and the end of that sigma is located where that -10 box is. So the sigma kind of identifies where these regions are on our big, DNA strand here, and this then helps our RNA polymerase know where it needs to connect, right? And so once this has happened, we'll be able to see that it will be able to move on and know where to start transcribing the gene. All right, so what's going on inside of the holoenzyme? So again, the holoenzyme includes sigma and the RNA polymerase. So again, it's positioned around that -35 and -10 box that we just looked at. And that sigma, again, can only bind in one direction. And so the orientation of the promoter then determines which of the two DNA strands will be used as the template, and then in which direction the RNA polymerase will be able to move. So if we go back here, you'll notice, it's looking at this strand, in this case up here, knowing where it needs to, which strand it's going to be able to transcribe. Unlike when we do DNA replication, where we had to use the enzyme DNA helicase to open up our double-stranded DNA helix, when we're only dealing with transcribing a gene, we're going to see that an RNA polymerase actually has that function. So this RNA polymerase is able to create what we're now going to call a transcription bubble that will open up the double helix so that we're able to get access to the template strand so that we can start building the messenger RNA. So the incoming nucleotides will enter through a channel in the enzyme and eventually get to the active site. So if you remember, and we'll look at it here shortly again, that RNA polymerase is a relatively large enzyme that had multiple points of entry in it. And so we're going to look at how we thread this template strand through, how the nucleotides are able to enter and actually interact with that active site so that we can start the polymerization process. Okay, so if we look over here on the left, we see our sigma molecule bound, and we see here our RNA polymerase enzyme. And you can see it's got these multiple points of entry into this actual enzyme. And so what happens is after this thing attaches, we're going to see that we're going to thread this DNA molecule through this opening here, right? So you see the DNA coming in, and the RNA polymerase then is able to open up this DNA molecule, and this then is going to function as our template strand. So you can see there's pieces inside the enzyme that's helping to direct this. And as we get to this active site, we're able to start adding these nucleotides as we kind of feed this strand through the opening here. All right, so yes, I might have misdirected. So it kind of bends through this channel here. So we're going to continue, the DNA is going to kind of move here from your right to your left and kind of get pulled through this enzyme, and we'll keep adding these nucleotides. So this picture over here where it says step 3 is kind of showing us that. So the enzyme's going to travel down the length, or we're going to pull this DNA molecule through. You can see that we keep adding these RNA nucleotides in, and then coming out through this doorway, we have our RNA, messenger RNA strand coming out. So a pretty little complex little machine there. So RNA polymerase is a pretty complex little machine that's doing a lot of action. So it's opening up the DNA, it's attaching it, it's reattaching it after it goes through. All right, so that's kind of the elongation step. So we just went through that kind of fast. So this is step 2 is elongation. It's just going to keep adding these nucleotides on. And then how does it know when to stop, right? So these genes can't go on indefinitely, and so the process needs to stop. And let's see. All right, so looks like we're just about, I'm a couple minutes over. I apologize for that. So we can pick up with the termination step, I guess, on Friday, and then we'll work on the worksheet. So that worksheet that I showed you guys at the beginning of class, we'll go ahead and work on that at the beginning of Friday's lecture. All right, so I'm going to go ahead. I want to respect your guys' time and stuff. So it is 12:54. I did run over. But if you have any questions, I can certainly stick around here for a few minutes and help you out. And again, I will hold class on Friday for those of you who want to attend. If you can't, it's absolutely no problem at all. I will record this and get it posted for you so you can watch it. Okay, so if anybody has any questions or anything like that, you're all unmuted, you can talk. So I hope you guys have a great weekend. I hope you guys have some time to enjoy, if you celebrate Easter, that you have time to do that somehow. I know all of my family's up in Ohio, so maybe we'll get to do a Zoom meeting with them or something. But we'll take care of that. All right, so those of you who want to stick around, there are a couple questions. So DNA polymerase 2. No, we did not go over DNA polymerase 2. So for what we need to know for DNA replication, we only needed to focus on DNA polymerase 3, which is the workhorse that lays down all of the nucleotides, and DNA polymerase 1, which is the one that replaces your RNA primer and puts down the new DNA nucleotides. So no, we did not discuss polymer 2. Good question. Good observation there. Anything else? All right. Well, I hope you guys all have a wonderful rest of the week. I hope I get to see some of you on Friday, and we'll take it from there. Bye.